Hi everyone. In this video, I'm going to show you how to make an agarose gel and then use electrophoresis to analyze and even purify fragments of DNA. But let's start off by talking about what an agarose gel is, how it's made, and how we do electrophoresis. So first of all, to make an agarose gel, you'll need a certain amount of agarose powder and some TAE buffer. So agarose itself is a polysaccharide, but it's unique in that it cannot be digested by bacteria. So that's why we use it to make LB agar plates, for example. Whenever we want to isolate bacterial colonies, the bacteria will grow on it, but they cannot eat it like other polysaccharides, like dextrose or Jello, for example. What we're doing today is very much like making Jello, but we're using a different sugar that bacteria cannot metabolize. Not that we're going to have bacteria on this agarose gel that we're making today, but it is just a very common staple in most labs, so that's why it's used for agarose gel electrophoresis of DNA here. Okay, so we need some agarose. The amount of agarose that we need depends on what type of gel we're running. Now, usually, you're going to run a 1% agarose gel. That concentration of agarose in the gel is sufficient to separate most types of DNA, most sizes of DNA fragments. Anything between, let's say, a few hundred bases to a few thousand bases. But if you're separating or analyzing a larger fragment, something like 10,000 bases or more, you might want to decrease that concentration from 1% down to 0.5%. Likewise, if you're analyzing smaller pieces of DNA, then you might want to increase the concentration up to, let's say, 2%, for example. But let's say we want to make a 1% gel, and we've got a gel cast that accommodates 75 ml of gel. So in that case, 1% of 75 ml would be 750 milligrams. Now for the buffer that I'm going to dissolve the agarose in, we typically use TAE buffer. There's other buffers like TBE, but the most common is TAE. That's tris acetic acid EDTA buffer. Now it's very important that I dissolve the agarose in TAE. I cannot dissolve it in ultra pure water because we're going to be doing electrophoresis, which means we're going to be passing an electrical current through this gel. If I pass an electrical current through pure water, there's a lot of resistance to that current because there's no electrolytes. And what that means is that gel made with pure water will get very hot. And that might actually ruin the gel completely. But if I use TAE buffer to make this gel, then there's electrolytes in there that can pass an electrical current through more easily. So it means it won't get as hot and you'll get a better gel overall. So just be very, very careful. Make sure you dissolve your agarose in TAE, not in ultra pure water. Okay, so anyways, we're going to dissolve the agarose in TAE, but that's not quite as simple as it sounds because agarose has a very low solubility in water. So what we have to do is we have to first heat it up in the microwave for a few minutes. And what that will do is, as we increase the temperature, we also increase the solubility of the agarose. So once it's boiling, pretty much all of the agarose dissolves. And then what we can do is we can add some ethidium bromide to that solution as well. So ethidium bromide is a fluorescent dye that binds the DNA, so that's going to let us see the DNA in the gel at a later step. But anyways, once we have the molten agar, we add the ethidium bromide, we let it cool down to room temperature. And what's going to happen there is as the temperature decreases, so does the solubility of the agarose, such that a gel forms. The agarose comes out of solution to form this gel that's mostly water, but you can pick it up like a solid. Anyways, once we have that gel, we also put it in a special cast that makes these wells here. So these would be hollow wells, these rectangles shown at the top of the gel. And that gives us a place to put our samples. So we can put our dark blue sample here in this well and this one. And then we can expose the entire gel to an electric field. Now what's going to happen here is if I have negatively charged DNA in this well, and I put the positive end of an electric field down here on the opposite side, the DNA will migrate towards the positive end of that electric field due to its charge. And what you'll find here is that smaller pieces of DNA, shorter pieces of DNA, will migrate through that gel faster than larger or longer pieces of DNA. simply because they're so bulky, it's hard for them to navigate through this gel here. And so we can use this type of phenomenon to separate pieces of DNA based on their molecular weight. So let's say you're running a PCR reaction. You get ample amounts of this PCR product, but you need to separate it from the template DNA. Let's say it's a plasmid that you used for that PCR. An agarose gel allows you to do just that. Because the PCR product is presumably smaller than the template you were using, it will migrate through faster. It will end up down here, whereas the template is up here. So then all you need to do is cut out this band down here, which is fluorescing because it's bound to ethidium bromide, and now you've got a purified fragment of DNA. So there you go, from start to finish. That's how we make an agarose gel, and that's how we use them as well. So now let's take a look at what this actually would look like in the lab. So first of all, we're going to start by making the agarose gel itself. Now here, you need to weigh out some agarose powder, so start by getting a weigh boat, put that on an analytical balance, and tare it. Then you want to take a scupula, weigh out the necessary amount of agarose powder. So again, let's say I'm making a 1% gel, and the volume of that gel is 75 ml. I would need 750 milligrams of agarose powder here. Now don't worry too much about getting that amount exactly perfect. Most bands will run just about the same if it's a 1% or even a 1.01% gel. And then I'm going to add that to the TAE buffer that I already have measured out here. And you'll notice initially it's very cloudy. So next I need to dissolve the agar. So I'm going to heat it here for about a minute, let's say. But the amount of time you need to microwave it's going to depend on the volume of gel you have. So you can see here, after about 30 seconds of heating, I haven't dissolved all of my agar yet. I still have a lot of white cloudiness there. So I'm just going to continue microwaving it until I get something that looks like this. A clear solution. You can see all of the agarose is dissolved into the solution. Once I am at that point, the next thing I'm going to do is I'm going to clean out my gel cast. So what you want to do here is you want to wipe everything down with ethanol, with the cast and the comb, just to make sure there's no DNA left over from the last person using the gel, or even just some dust or bacterial contamination that could have happened to land here on the cast. So ethanol is a great way to clean those things off and make sure that you don't have any other contaminating DNA in your sample. So you want to place your well comb into the gel cast, and then remember the gel is going to be very hot here, so you want to use tongs or a handle to pick up the flask and pour the gel into the cast. Take special care here to make sure you don't make any bubbles in the gel, as they can interfere with the migration of DNA through the gel. Now the next thing I'm going to do is I'm going to add my dye here. In this case it's ethidium bromide. I'm going to add that directly to the molten gel while it's still a liquid. So I'm going to try to spread that around with the pipette, and then I'm going to shake the gel a little bit to make sure that that ethidium bromide is perfectly dispersed. You want to make sure that it's very spread out, because if you have a lot of ethidium bromide in one spot, your gel will be blotchy. Now the other way you could do this is to add the ethidium bromide first. You could add the ethidium bromide to the cast first and then pour the gel on top. Either way you do it, just make sure that the ethidium bromide is spread out in the gel. You really shouldn't be able to see it. It should be so spread out. I would like to take this opportunity, though, just to remind you, ethidium bromide is a carcinogen. Just like it binds the DNA in the gel, it can also bind to your DNA. So if it gets into your cells, it could bind some DNA. If it binds at the wrong place at the wrong time, that is something that could be oncogenic, something that could cause cancer. So I would definitely recommend here, be very cautious when using the ethidium bromide, and whenever you work with it, I would get new gloves as soon as you can. If your gloves have touched the gel, for example, that contains ethidium bromide, I would discard those gloves and get new ones, just for the sake of caution. Okay, so once you have that gel poured, you're going to wait probably around 20 to 30 minutes, longer times for larger gels, for that gel to solidify. But once it solidifies, it should look something like this. Kind of hard to tell in the image, but it's going to have kind of a semi-opaque look to it. It's going to be whitish gel. But you want to give it ample time to solidify. you don't want to pull the gel out or the comb out too early because then you'd have to start over again. You can always poke the gel with your finger just to make sure that it feels solid as well. So anyways, while that gel is solidifying, another thing you can do is you can clean out your bath. So just like we did with the cast, we're going to clean out the gel bath with ethanol as well, just to make sure there's no contaminating DNA or other things in that gel bath. But once you have that clean, you're going to remove the comb. Make sure you lift that comb slowly and straight up to make sure you don't tear the barriers between the wells. If your gel is too long for the gel bath, you can go ahead and cut it here. And then you want to loosen the cast and remove the gel. You want to transfer that directly to the gel bath here. Slide it in with your fingers. And then if you cut off any gel, make sure you dispose of that in the lithium bromide waste. Next, just like we made the gel with TAE buffer, now we're going to submerge the gel in TAE buffer. Again, because we are passing an electric current through this, we need the electrolytes in TAE. Do not submerge the gel in pure water. It will get way too hot. Okay, so you can see there we've submerged it. So now we're ready to prepare our samples. Now for that, we add loading buffer. So with loading buffer, you want to add a certain amount to your sample. Different loading buffers come as different stock concentrates, 2x, 5x, 10x. Check your specific loading buffer and see how much you need to add. But we're going to add that to the solution, the sample, and then we're going to pipette up and down to make sure that's nice and well mixed. Now what we're doing here is we're adding glycerol and a loading dye to that sample. The glycerol is going to keep the sample in the well. The loading dye is going to give us something that we can track as it moves through the gel. Alright, so now I'm ready to load the gel. The first thing I'm going to load is a DNA ladder. Now this is a sample that contains DNA fragments of known length, so it's going to be a reference for us. It's going to allow us to see where a fragment of 100 bases, where a fragment of 1,000 bases is in the gel. Alright, so once I load that into the gel, I'm going to load my sample. Now if your sample is large, you might have to load it into 2 separate wells like I'm doing here. But the trick to loading these gels is to load them slowly. The slower you go, the less chance it's going to be that the sample is going to leap out of the well. You also want to inject the tip just barely into the well. You definitely don't want to inject the tip too far into the well, because then you might actually poke through the bottom of the gel. So you want to be on the top of the well, slowly eject your sample, and that's all there is to it. By all means, use 2 hands if you need to. Alright, and then we're going to put the lid on the gel bath. Now here, make sure that you've got the positive end, the red end, on the far end of the gel. And then we're going to set the power supply to 110 volts and let that run for about 15 to 30 minutes. But it depends on what your product is. Now as soon as you turn on that power supply, you should see bubbles coming up from the electrodes. That's the electrolysis that's happening because we're applying an electric field. If you don't see bubbles, something is wrong. Okay, so this is what your gel should look like after about 15 to 30 minutes. You can see here the dyes that we had in the loading buffer have migrated through the gel. So I can see where my DNA band should be, somewhere between those bands. So that bottom band, for example, would correspond to a very short fragment, something like 100 to 200 bases of DNA, whereas the top band in this loading buffer corresponds to something that's a few thousand bases long, something like a plasmid. So you want to look at these bands and make sure you run the gel to where they are sufficiently separated, but you don't want to run the gel so long that one of these bands actually falls off the gel. Okay, so you could definitely take, here I've only used about half of the gel because I really am separating just 2 fragments, a smaller PCR product from a very large template DNA. So I don't need to use the whole gel, but if I had 2 fragments that were closer in size, let's say 500 and 1,000 bases, I might want to run this gel for considerably longer to ensure that I had proper separation between those bands so that I could definitely see them and distinguish them. And if I'm purifying one of those bands, I need to have them far enough apart to where I can cut them out and not have my spatula touch the other band while I'm trying to isolate one band. Okay, so once you have that gel, the next thing we're going to do is visualize the DNA. You won't be able to see the DNA with your naked eye until you put it onto a UV visualizer like this. Okay, so I'm going to put the gel on, and then remember, UV light is also carcinogenic, so I'm going to put a plexiglass cover on top of that gel before I turn on the UV light. I'm going to turn off the lights in the room just to make sure I can see the bands as clearly as possible. Then I'm going to turn on that UV light here, and this is an example of what a gel might look like. Now in this specific gel, unfortunately my ladder was degraded, so it doesn't look all that great, but I can actually see my PCR product that I'm running in this gel. As you can see, I've got two nice bright bands there. Now this is what that gel should have looked like, so just to update this video, the ladder should look like this. You can see I've got multiple bands here that I can clearly differentiate, and I know from the manufacturer that the bottom band is 500 bases, then we've got 1,000 bases on top of that, 1.5, 2,000 bases, and then there's this very bright band. There's usually a brighter band in the ladders just to help you distinguish them. This one is 3,000 bases. Okay, so in this case, I was hoping for a PCR product that was around 950 bases. And what I see over here in my samples are three different bands. The lower bands would be unused primer. So primers, even though they're single-stranded, sometimes they can form double-stranded secondary structures, or they can bind to one another, or just bind to ethidium bromide non-specifically. So you will see some smearing sometimes for primers that are down here, even though they're single-stranded. But up here, I can see I have a very bright band that corresponds to my PCR product. So there you go. And I can estimate the size of this product by looking at the ladder. I know very crudely that it's between 500 and 1,000 bases, but you can see it's closer here to 1,000. So I feel pretty good about this product being around 950 bases because it's so close to that rung on my DNA ladder. And then unfortunately it looks like I've got a little bit of off-target amplification here too. I've got another product from the PCR reaction that's about 1,500 bases. But I've run this gel long enough to where I can easily cut this band out and leave that other unwanted band in the gel. So there you go. From start to finish, that's how you make and run and analyze an agarose gel. Just once again, please be very careful with ethidium bromide and ultraviolet light, both of which are carcinogens, so use the appropriate protective gear whenever you're working with them. And definitely with ultraviolet light. There will be times, especially when you're needing to extract pieces of DNA from the gel, make sure you're wearing a visor over your face to protect yourself when you take off that plexiglass cover from the UV transilluminator, and make sure you've got long sleeves and gloves on to protect your skin as well. One final note would be that just like UV light is a carcinogen for you, it can also mutate your DNA samples as well. So these bands that we have here in the gel, UV light can also damage them. So whenever you're visualizing your samples, if you're going to be using them for a downstream application, let's say cloning this sequence into a plasmid, you want to make sure that you work with UV light as quickly as possible. So you want to minimize the amount of time that you're illuminating these bands with ultraviolet light to minimize the chances that they will be mutated by that high energy light. All right, so there you go. Good luck with your gels. I hope they turn out great, but do let me know if you have any problems. Sometimes you do need to troubleshoot these gels. Your bands appear blurry or something like that. You can look online or just email me directly for assistance. All right, good luck. I should also add that the gel image you're seeing here appears orange instead of the pinkish purple in the other image because I'm using a photo filter to capture this image here. If you just use an orange photography filter, it will block out the UV and bluish purple light that we get from the UV lamps, and will only let through the ethidium bromide fluorescence, which appears orange in this figure. So it just makes for a cleaner image.